what if the very drug designed to cure your cancer is exactly what teaches it how to survive?
Yeah, I mean, it is a deeply unsettling medical paradox for sure.
Right. And today we are jumping into a deep dive on exactly that, because for decades, if you were diagnosed with advanced stage 4 colorectal cancer, the medical community offered their absolute best, heaviest hitting chemotherapy treatments.
They really did. But despite throwing our most toxic chemical weapons at the disease, the 5-year survival rate has just, well, it's stubbornly sat at a grim 12%.
12%. I mean, that is just a brutal statistic.
It is. And for the longest time, we thought we knew why. We just assumed the cancer cells were simply mutating randomly, developing these hard-coded genetic shields against our drugs in a classic survival of the fittest scenario.
Which makes sense on paper. You find the broken gene, you target it, and if the tumor comes back, you just assume it acquired a new random mutation.
Exactly. That was the prevailing dogma. We viewed cancer almost exclusively through the lens of genetic mutation. But there is this groundbreaking 2026 paper published in Nature that just completely shatters that assumption.
Oh, it really does. And the researchers basically proved that the tumors aren't just mutating, they are quite literally shape-shifting.
Yeah, which is terrifying but also fascinating.
Okay, let's unpack this. Because the mission of our deep dive today is to explore this biological superpower, which scientists call cellular plasticity. We're going to look at how a tumor can physically change its state to survive.
And how our standard therapies are accidentally forcing it to put on biological armor.
Right. And then, most importantly, we're going to talk about how a brand new algorithmic 1-2 punch strategy could fundamentally change the odds for that 12%.
Because to defeat a shapeshifter, you really must first deeply understand its forms. And what this paper outlines so beautifully is that tumors are not just a uniform mass of identical cells.
They're an ecosystem.
Precisely. They are a dynamic ecosystem. Specifically, the researchers found that these colorectal tumors constantly flip back and forth between two distinct states.
Which I've been calling the seesaw of survival in my head just to visualize it. So on one side of the seesaw, we have a cell state marked by a protein called LGR5.
Right. And on the other side, we have a state marked by TROP2. Let's break down the mechanics of that seesaw, starting with LGR5.
Okay, lay it on me.
So in healthy biology, LGR5 is the traditional marker for adult stem cells in your intestines. But in a tumor, it marks the cancer stem cells. The defining characteristic of these LGR5 cells is that they have incredibly high WNT signaling.
And WNT, which is spelled WNT if you're listening, is essentially a biochemical pathway.
Yes, exactly.
Like if we think of a cell as a car, the WNT pathway is basically a biological accelerator pedal, right?
That is a perfect analogy. When WNT signaling is high, it tells the cells to grow, divide, and rapidly build the sheer bulk of the primary tumor. They are the factory workers of the cancer, multiplying relentlessly.
Okay, but sitting on the exact opposite side of that seesaw is the TRP2 state.
Right, which stands for trophoblast cell surface antigen 2. And these cells are the exact opposite of the LGR5 cells.
How so?
Well, they have very low WNT signaling, so they basically take their foot completely off the accelerator pedal. Instead of acting like rapid factory workers, they adopt what researchers call a fetal-like state.
Wait, fetal-like? Meaning they revert to behavior from when the human body was just an embryo.
Precisely. Think about what embryonic trophoblast cells have to do. They have to aggressively invade the uterine lining to implant an embryo.
Oh, wow. So these cancer cells reawaken that ancient primal programming.
Yes, they become high relapse cells. And when the researchers analyzed a massive cohort of over 1,300 colorectal cancer patients, the clinical data was striking.
What did they find?
High TRP2 expression directly correlated with distant metastasis and poor disease-free survival.
Meaning the TRP2 cells are the ones that actually kill the patient by spreading the cancer to other organs.
Yes. They are particularly dominant in notoriously treatment-resistant tumors. But here is the critical mechanical question, why?
Well, wait, that's exactly what I wanted to ask. Because on the surface, this feels, I don't know, a bit counterintuitive.
How do you mean?
Well, if LGR5 represents the rapidly dividing stem cells that literally drive the bulk growth of the cancer, why are these slow, WNT-low TRP2 cells the ones initiating the deadly metastases? It seems like the fast-growing cells would be the most dangerous.
What's fascinating here is the physical reality of what it takes for a cancer cell to metastasize. A rapidly dividing LGR5 cell is great at building a tumor locally, safely anchored in the colon.
Right, it's safe at home base.
Exactly. But a cell's journey through the bloodstream is violently hostile. First, when a normal cell detaches from its structural home, it triggers a biological suicide program called anoikis.
The cell literally self-destructs.
It does. It just self-destructs. So just by detaching to enter the blood, the LGR5 cell is already fighting its own self-destruct code.
That's
And second, the sheer stress of blood flow, like just the physical force of your heart pumping, can tear a fragile, rapidly dividing cell into pieces.
ouch. Okay.
And third, because LGR5 cells are highly active and loud, biologically speaking, your immune system's macrophages easily spot them and wipe them out.
So the LGR5 state is basically a death sentence if they enter the bloodstream.
It is. So to survive the road trip, the tumor cells actively drop their LGR5 expression. They pull back on the WNT accelerator, stop dividing, and adopt this fetal-like TROP2 state.
And this state acts as like heavy, stealthy armor.
Exactly. It shuts down the self-destruct sequence, reinforces the cell against physical shear stress, and allows them to sneak right past the immune system to seed new micrometastases in distant organs.
Like the liver.
Right.
That is just terrifyingly elegant. I mean, the cell literally changes its entire identity, pausing its own growth just to survive the journey.
It's an incredible survival mechanism.
Okay, we are a few minutes into this deep dive, so let's pause and lock down the core mechanics of what we've covered so far with a quick recap.
Sounds good.
Point 1. Tumors are not one uniform thing. They rely on cellular plasticity to seesaw between two states, the rapidly dividing LGR5 cells and the stealthy, slow-growing TROP2 cells.
Spot on.
Point 2. TROP2 is the armored state responsible for the tumor spreading to other organs, which is the primary cause of poor prognosis.
Yes.
Which brings up an interesting question for you listening to consider. If you were trying to cure this disease, knowing what we just discussed, would your instinct be to target the actively dividing cells, making the primary tumor bigger, or the sneaky traveling cells that cause the fatal spread?
That is the exact dilemma that leads us right into the paradox of our current standard of care. Because for microsatellite stable colorectal cancer, which accounts for about 90% of all cases, the foundational treatments are cytostatic chemotherapy regimens.
Right. In the clinic, you'll hear these referred to as FOLFIFAR or FOLFOC.
Exactly.
And for those not in the oncology world, FOLFIRI and FOLFOXIS are essentially cocktails of incredibly potent chemical toxins. They use drugs like 5-fluorouracil combined with either irinotecan or oxaliplatin.
Which are very aggressive.
Yeah, and the whole philosophy behind traditional chemotherapy is to just carpet bomb the body with chemicals that selectively kill rapidly dividing cells.
Correct. For example, 5-fluorouracil acts as a booby trap. It masquerades as a normal building block of DNA.
Oh, flaky.
Very. When a fast-growing cell tries to copy its genetic code to divide, it accidentally uses the chemotherapy drug instead of the real nutrient. This breaks the DNA strand and kills the cell.
Okay, so by definition, these standard chemotherapy drugs are perfectly designed to target and kill the fast-growing LGR5 cells.
They are. But here is the shocking discovery the researchers made. They grew patient-derived organoids in the lab.
Which are essentially 3D mini-tumors grown from living tissue taken directly from patients, right?
Yes. And they also used genetically engineered mouse models. When they hit these living tumors with the standard FOLFIRI chemotherapy, the drug did exactly what it was supposed to do. It successfully wiped out the LGR5 cells.
But, and I feel a big but coming.
But it simultaneously induced a massive sweeping surge in TROP2 expression across the surviving tumor cells.
Wait, so if I'm hearing you right, the treatment meant to cure the cancer is actually triggering the exact state that makes the cancer metastatic?
That is exactly what the data shows.
But wait, I need to push back on this a little. Is this actually cellular plasticity, or is this just basic Darwinian natural selection?
That is a very important distinction to make.
Right, because if you dump a toxic chemical on a tumor and it kills all the LGR5 cells, wouldn't the few pre-existing TROP2 cells just survive because they're slow growing and then just multiply once the chemo is gone?
That was the big question, and it's exactly what the researchers had to prove. To figure out if it was just natural selection, killing the weak and leaving the strong, or active Lamarckian-style shapeshifting, they deployed a brilliant genetic lineage tracing tool.
Oh, this is the genetic tripwire experiment. Explain how this works, because it's honestly fascinating.
So they engineered mice with a very specific genetic modification called TAXD2-CRE. They set up a system where the exact moment a cell turned on the gene to build TROP2 armor, it would also permanently snip a piece of its own DNA, causing the cell to glow fluorescent red.
That's brilliant. It's like throwing a permanent dye pack on a bank robber.
Yeah.
Even if they take off the mask later, they are permanently covered in red dye.
That is a great way to think about it. It allowed them to track the history of individual cells. So when they subjected the tumors to chemotherapy, they watched cells that had never been TROP2 before, cells that were not red, suddenly flip their genetic switches and turn bright red in real time.
Wow. Proving conclusively that the chemo wasn't just selecting for pre-existing TROP2 cells.
Right. The chemo itself was the environmental stressor forcing the state shift.
Yes. The toxic stress of the therapy actively forced the surviving LGR5 tumor cells to panic and pull the TROP2 emergency lever. The therapy was driving the plasticity.
It's a staggering realization.
We've essentially been throwing a punch that teaches the opponent how to dodge perfectly.
Exactly.
Okay, time for our second checkpoint to synthesize this. Point 1. Standard chemotherapy regimens, the bedrock of our current treatment, successfully target the rapidly dividing LGR5 cells.
Check.
Point 2. The tumor actively responds to this toxic attack via cellular plasticity, drastically increasing its TRP2 expression to put on that fetal-like armor and survive.
Yes.
Which really makes you think about how we approach problem solving in general.
Yeah.
Have you ever implemented a major fix for a problem, only to realize your fix just forced the problem to evolve into something much harder to catch?
It is deeply frustrating. But in science, uncovering exactly how a system fails is often the prerequisite for learning how to control it. Because if we know for an absolute fact that the tumor is going to panic and put on its TROP2 armor when we introduce chemotherapy, we can proactively design a weapon specifically meant to pierce that exact armor.
Which brings us to the targeted assassin. We're talking about a completely new class of biological weapons called antibody-drug conjugates, or ADCs.
Yes. And in this study, they primarily use an ADC called sacituzumab govitecan.
Or Trodelvy in the clinic. Now, to help visualize how these ADCs operate, I like to use a simple mnemonic. ADC stands for Address, Delivery, Combat. It's a 3-part system.
I love that. It's a highly accurate way to break it down. Let's look at the mechanics, starting with the address.
Okay.
The ADC contains a laboratory-engineered humanized monoclonal antibody. You can think of this antibody as a highly specific GPS locator. It constantly circulates and scans the surface of cells until it finds the exact molecular shape of the TROP2 glycoprotein.
And it ignores everything else.
Completely ignores everything else. But once it finds that specific TROP2 address, it docks.
And that brings us to the delivery phase.
Right. Once docked, the cancer cell, thinking this is just a normal biological interaction, essentially swallows the ADC, internalizing it into its own cellular body.
And once it's locked safely inside the enemy walls, we get to the combat phase. The ADC releases its hidden payload.
And the payload here is devastating. In the case of sacituzumab govitecan, the payload is a chemical called SN38,
which is an incredibly potent topoisomerase inhibitor. Now, we need to explain topoisomerase because this blew my mind when I first read it. DNA is tightly coiled up inside a cell, right?
It is. It's twisted into incredibly dense structures. Whenever a cell needs to read its DNA or copy it, it has to untwist it first.
Right.
Topoisomerase is the essential biological enzyme that carefully cuts, untangles, and re-glues the DNA so it doesn't get hopelessly knotted.
So when the SN38 payload gets released inside the cell, it physically disables that untangling enzyme.
Exact.
So when a TROP2 cancer cell tries to do literally anything, its own DNA gets hopelessly knotted and it essentially shreds its own genetic code from the inside out.
It leads to immediate targeted cell death. But we have to remember the ecosystem we're dealing with. We talked about the seesaw of survival, right?
The tumor is plastic.
Yes. When you deploy the ADC and start slaughtering the TROP2 cells, the broader tumor ecosystem notices the change in the environment.
And the researchers didn't just guess what happened next. They tracked it using some truly futuristic technology. They used single-cell RNA sequencing combined with an algorithm called Palantir pseudo-time trajectory.
Single-cell RNA sequencing is revolutionary. Traditionally, sequencing a tumor was like putting a whole fruit smoothie into a chemical analyzer. You could tell generally what was in it, but the details were blended together.
Right, just a mush of data.
Single-cell sequencing allows us to unblend the smoothie. We can look at the exact genetic programs running in thousands of individual distinct cells simultaneously.
And then they feed that massive amount of individual data into the Palantir pseudotime algorithm, which the best way I can describe it is like a cellular time machine, or maybe a predictive physics engine.
That's a good way to put it.
It analyzes the RNA states of all these cells and calculates the mathematical probability of what biological state they will transition into next.
Precisely. And when they mapped the tumor's response to the TROP2-targeted ADC using this algorithm, they witnessed a second massive state shift.
What happened?
As the ADC wiped out the TROP2 cells, the surviving tumor cells reacted to the new environmental pressure. To escape the targeted assassin, they dropped their TROP2 armor and frantically shifted back into the fast-growing LGR5 state.
They jumped right back to the other side of the seesaw.
They did.
But wait, if the ADC kills the armored cells and forces the survivors back to the LGR5 state, doesn't that just recreate the exact same rapidly dividing primary tumor we started with?
It does, which is why the researchers noted that the ADC administered on its own is not a permanent cure. The tumor simply uses its plasticity to shape-shift away from the drug, hiding in the LGR5 state where the ADC's GPS locator just can't find it.
Wow. Okay, let's do our third checkpoint. Point 1. Antibody-drug conjugates, or ADCs, act like targeted smart bombs. Address, delivery, combat. They seek out the TROP2 armor and destroy the cell by shredding its DNA from the inside.
Perfect summary.
Point 2. Through single-cell sequencing and predictive algorithms, we can see that destroying TROP2 cells forces the tumor's plasticity into reverse, reverting it right back to the LGR5 state.
Correct.
So this raises the ultimate strategic question for you listening. Now that we know exactly how to push the tumor back and forth, chemo pushes it to TROP2, ADCs push it to LGR5, how can we use this seesaw against it?
This is where we arrive at the grand synthesis of the paper. For decades, oncologists viewed cellular plasticity as the ultimate unbeatable mechanism of drug resistance. It was the frustrating reason tumors always seemed to outsmart our therapies.
But the researchers realized they didn't have to beat the plasticity, they could exploit it.
Exactly. Instead of treating the tumor like a static target, they designed an algorithmic 1-2 punch combination therapy.
Okay, here's where it gets really interesting. Walk us through the algorithm.
Step 1 of the algorithm. You hit the tumor with the standard FOARI chemotherapy. As we established, this acts as an extreme environmental stressor. It wipes out the LGR5 cells, and it forces the entire surviving tumor mass to frantically shapeshift into the fetal-like TROP2 state to survive the chemical assault.
You are purposefully hurting them. You are forcing the entire tumor to put on the armor.
Yes. Step 2. You wait for the tumor to fully commit to the TROP2 state. And while it is completely trapped in this highly vulnerable, slow-growing state, you unleash the ADC, the sacituzumab govitecan.
Oh, man.
Because the initial chemotherapy forced nearly every surviving cell to upregulate TROP2, the ADC now has an overwhelming number of targets to hit.
It's basically an ecological ambush.
It really is.
But wait, I have to ask about patient safety here. This sounds incredibly aggressive. If we hit a patient with a massive dose of standard, highly toxic chemotherapy just to herd the tumor, and then immediately hit them with a potent antibody-drug conjugate, isn't that incredibly dangerous? How does a human body survive a biological 1-2 punch that intense?
That is a vital clinical concern. Toxicity management is everything, but the brilliance of this algorithmic approach is that it is sequential, not simultaneous.
Oh.
You don't deliver both massive toxic loads at the same time. You deliver the chemotherapy, which pushes the tumor into the TROP2 state. Because TROP2 cells are slow-growing and essentially in a state of suspended animation, you have a therapeutic window.
So you can pause.
Right. You let the patient's healthy, rapidly dividing cells like their hair follicles and gut lining recover from the chemo. Then you administer the ADC. And remember, the ADC is highly targeted. It only releases its toxic payload inside cells that display the TROP2 address, largely sparing the patient's healthy tissue from the second blow.
That makes total sense. And the physical evidence for this algorithmic approach is incredible. The researchers used patient-derived organoid xenografts, or PDOXs.
Yes, meaning they took a patient's lab-grown mini-tumor and surgically transplanted it into a living mouse to observe the real-world vascular and biological response.
And what happened?
The results were undeniable. When mice with either early micrometastases or established macrometastases were treated with this specific sequential combination of standard chemo followed by the ADC, the researchers measured vastly reduced bioluminescence.
Right, and just as a reminder, Bioluminescence is a tracking method where they genetically alter the tumor cells so they literally glow. The dimmer the glow, the less tumor volume remains in the body.
Exactly. The tumors shrank dramatically. But more importantly, the survival rates of the mice on the sequential combination therapy significantly increased compared to mice given either the chemotherapy alone or the ADC alone.
That is amazing. If we connect this to the bigger picture, it truly represents a paradigm shift in how we think about treating disease.
It really does.
It's kind of like dealing with a terrible pest infestation in your house. Instead of just spraying chemicals blindly in every single room, you drop the temperature in the house so all the bugs are forced to hide in one specific corner to stay warm.
I love that.
And then you just fumigate that one corner. We are using the cancer's greatest evolutionary trick, its plasticity as a leash, to guide it directly into a therapeutic trap.
It is the absolute essence of adaptive strategy. We are finally speaking the tumor's language, manipulating its dynamic ecology, rather than just attacking it with blunt force.
Amazing. Let's do our final checkpoint before we wrap up today's deep dive. Point 1. Combining standard chemotherapy with TROP2-targeted ADCs in a sequential algorithm creates a devastating, inescapable trap for the cancer.
Yes.
Point 2. By deeply understanding the underlying mechanisms of cellular plasticity, we can actively manipulate the tumor, forcing it into a predictable and highly vulnerable state. Which leaves a great conceptual question to consider. Can you think of a situation in chess, sports, business negotiations, or even daily life where you use an opponent's greatest strength to predict and completely counter their exact next move?
That concept of exploiting an opponent's adaptation is exactly what is driving the cutting edge of oncology today.
It really is a marvel. I mean, we've moved from blunt force genetic attacks to strategic ecological management. Thank you for joining us on this deep dive into the world of cellular plasticity. I hope you carry this new lens of adaptive strategy into your own learning journey, whatever field you happen to be in.
And I would like to leave you with one final provocative thought to mull over. Today we talk specifically about exploiting directed plasticity in colorectal cancer. But if we can literally force cells to adopt specific, vulnerable states simply by changing their environment with drugs, could this principle of directed plasticity be the key to managing other evolving biological threats?
Oh, that's an interesting thought.
Right. Could we one day use an algorithmic sequence of drugs to herd autoimmune diseases into a permanently dormant state? Or could we trick antibiotic-resistant superbugs into dropping their defenses just before we deliver a fatal blow? The biological seesaw is out there. We just have to learn how to push it.